So this will be a brief overview as well as an outline for the Exercise 7 lab report on photosynthesis for Bio 111. We established that the background for photosynthesis is that the sun is the ultimate source of energy for all organisms on Earth, and it is the process of photosynthesis that converts that energy that's found in sunlight into a chemical energy that is usable by cells, which is a chemical energy that's in the form of carbohydrates or starches. And photosynthesis occurs specifically in three kind of major groups of organisms: the plants, the plant-like protists, and the cyanobacteria. Regardless of what type of photosynthetic organism it is, we see that the photosynthetic organisms are collectively referred to as autotrophs, because the term autotroph means self-feeding, because they are capable of producing their own carbohydrates or starches, which they will use in their own metabolism to make ATP energy. Whereas we also saw and defined heterotrophs, which are organisms that feed on others, and they will typically consume either autotrophs or other heterotrophs for their sources of energy, that is, for their sources of chemical energy of carbohydrates or starches. All of the photosynthetic organisms contain a green pigment called chlorophyll, and it's chlorophyll that essentially harvests and captures the energy from light and begins that conversion of the light energy into chemical energy. Figure 7.1 on page 80 summarized the equations for photosynthesis, and it also did a really good job of showing kind of where all of the chemical components from the initial part of the reaction end up in the final part of the reaction. We also saw in the background that photosynthesis can be divided into two reactions. We had a light-dependent reaction as well as a light-independent reaction. The light-dependent reaction, as its name implies, involves the chlorophyll pigments, and it involves them absorbing that light energy and beginning that initial conversion of light energy to chemical energy. In that reaction, we saw that water was split, creating oxygen as a byproduct. So the oxygen molecule or the oxygen atom in the water is what creates oxygen gas as a byproduct. We also see that some of the hydrogen from the water or the hydrogen from that water is used in the light-independent reaction, which we'll go over next. So next we saw the light-independent reaction, which is also called the dark reaction, the name is a little bit misleading because the light-independent reaction often does occur in the presence of light. It's simply a reaction that does not require light in order for it to occur, but it often does happen when light is present. So it doesn't always occur when the plant or the photosynthetic organism is in the dark. In the light-independent reaction, we see that chemical energy from the light reaction is used to make glucose. So this is the glucose building reaction because the light-independent reaction uses carbon dioxide from the atmosphere because the carbon that's in glucose ultimately comes from the carbon dioxide that's in the atmosphere, as well as the hydrogen that was split from water in the light-dependent reaction. We saw that the chloroplast was the organelle that contains the different chlorophyll pigments that are used in photosynthesis. And we also saw that the lab manual gave us the two hypotheses that we're using in our photosynthesis lab report, because we essentially completed two experiments in that. So we'll have two hypotheses, and we'll kind of approach the lab report as two separate experiments kind of rolled into one. The first hypothesis was that it was hypothesized that carbon dioxide is not necessary for photosynthesis, so we can use that. You can use that one word for word. You can just remove the quotations there. The second hypothesis we're going to test is that all chloroplast pigments have the same solubility. And again, you can use that word for word and remove the quotations there. So moving on to the results section, you can kind of split the results section into essentially two pieces or two halves. One part of the results section for each of the experiments. For the leaf experiment, where we exposed them to starch, what I would like you to do is include the photos of your leaf after you have stained them with the iodine solution. So you should have 2 photos, one of each leaf, or 1 photo with both leaves in it. Recall that iodine turns blue or a bluish black color in the presence of starch, which is our indicator that photosynthesis has occurred, because starches will be made when photosynthesis occurs, as we just showed in the light-independent reaction. Because the light-independent reaction is very much dependent on the byproducts that are made in the light-dependent reaction. We also saw that iodine will remain a kind of yellowish gold color if starch is absent, so that is our kind of visual indicator that photosynthesis did not occur in that particular experiment. So what I want you to just take a look at is your 2 leaves side by side and describe the results of that experiment with the leaves that you stained with starch. Of course, we saw that the leaves were held under 2 conditions. There was one condition where plant A was exposed to CO2 and plant B was exposed to an environment where CO2 was absent. So plant A was exposed to normal air with carbon dioxide in it under the bell jar, and plant B was exposed to air that had the CO2 removed by soda lime pellets. And so figure 7.2 in the lab manual on page 80 gives a good visual representation of that setup there. So as far as where your data analysis is concerned, what I want you to do is take a look at what you observed in both leaves with respect to the color after you stained them with iodine, and describe the results that you saw in the context of if that leaf showed the presence of starch, if it turned blue-black, or if it showed the absence of starch because it was mostly yellow or gold in color after staining with iodine. So therefore, figure 1 should be the image with both leaves in it, or 2 different images if you have a photograph of each leaf in separate images. And in the figure caption for figure 1, you should explain that they were stained by iodine and whether and kind of describe your specific results there, whether you observed the presence or the absence of starch in them, depending on what color it had turned after exposure to the iodine solution. And the results for experiment 2, which could be like a separate paragraph or a separate small paragraph with a separate figure, where we tested the experiment that tested the hypothesis rather that all chloroplast pigments have the same solubility. Please refer to that video that I sent by email to you a few days ago, because it has the data for that experiment and also has the instructions for creating a really simple bar graph of those data. So it's not a particularly long video. It should just take you a few minutes to get it all set, go through it, get it all set up, get the data from it, and use that in your lab report. So that brings us to the conclusion, right? So kind of very simply restate both of your hypotheses at the beginning of your conclusion section, and then from there you should state whether each hypothesis was either supported or not supported, and then use your data to explain why each of those hypotheses was either supported or was not supported, and then wrap that up by relating it to the background information. And of course, in your literature cited section, as well as in your background information, in the initial hypothesis section, as well as in the conclusion section, you can cite the lab manual as we've done before. So that's nothing there's nothing that's changed there. So you are welcome to use the lab manual citation. Just make sure to change the chapter name and page numbers as relevant in that literature cited section. And you're also welcome to use the OpenStax textbook as well. Just make sure to change the chapter name and number to reflect the actual chapter that photosynthesis is discussed in the OpenStax textbook.